can have a chance to have a watch. All right, let's go. What do you got for me?
I do have one question.
By all means, Jasmine.
So with strength gains, in the one RM and multiple RMs, I was having a harder time to figure out what exactly you wanted from us there.
I'm not, I don't, can you be more specific? Like what do you
Um,
mean? Like in you yes, you gain strength in all of those, but what else were you suggesting?
well,
I,
I'm,
I'm
I'm I'm asking what we should be studying for that. Like, I have what was on the slides, which is like greater and dynamic one RM actions compared to isometric, and then I have like the Hubel et al. Like little like statistics because I didn't know if you wanted those or not. But like sorry about my computer, it's going crazy.
Oh, that's
Uh,
okay.
but yeah, I'm not sure what exactly you want me to uh have like learn from that.
I mean, it really is mostly just that there are greater strength gains typically during dynamic contractions than during isometric during like MVCs because
Okay.
you get more accustomed to doing right when you work out. For the most part, you're doing some kind of like a dynamic, a concentric, eccentric. You're doing a dynamic contraction, so the adaptations are going to be greater when you test using something like that. Like that's the big kind of point in the take home. Okay.
Okay, that makes sense. I was just making sure there wasn't more to it that I needed to know
Oh,
because
no, no, there's not, at least in regards to those things. What else we got? Okay, sweet. That was quick. That was easy. We'll see y'all on Wednesday.
I had a
maybe.
question about, oh,
Oh, sorry, go
I don't
ahead.
know if you're, okay.
Go for it,
um,
Tyler.
One of the questions, or like the short answer questions, it was the first one of metabolism. It's how, explain how the ability of the electron transport chain to recycle NADH to NAD plays a role in the amount of lactic acid formed during exercise.
Hmm.
I was, I was kind of confused on how to answer that. I know that like the recycling of NADH to NAD like decreases the pH, and would that be,
So,
or is that wrong?
um, so I mean, there's a part of that that's right. So let's talk our way through all of this.
Okay.
Okay. This is one of those critical thinking questions where there's not sort of an easy, straightforward thing. All right. So let's take it in this way. So look at the question this way. Okay. What are the things, or what is the primary thing that regulates how much lactic acid we make? Let's start there. Before we can figure out the role that recycling NADH to NAD might play. Okay, when are we going to make lactic acid? Like, what's going to be the kind of conditions that are going to make that happen? Anybody can answer. It doesn't have to be Tyler.
Isn't it the absence of oxygen?
Okay, that is certainly that's a scenario, Montana, right? Um, but let's think about it this way then. If I don't have oxygen, why would I make lactic acid?
I mean, lactic acid is made whenever you have like an increase in H plus, so it's like the byproduct, right?
So that's probably backwards, Karen, but you're thinking about it the right way. So when you make the lactic acid, it then dissociates into lactate and the H plus and the hydrogens. So when one goes up, the other one is going to go up, but you actually have the kind of cause and effect of that. But why, if I don't have any, let's go this other way. Why, if I don't have any oxygen, would I make more lactic acid? Because this is going to, in some ways, help us get to the answer of our question.
Doesn't it have to do with like the electron, like it's the last acceptor? So like
Okay.
it's backed up.
Okay. So if I don't have any oxygen, I can't run the electron transport chain. So if I can't run the electron transport chain, what else is then not gonna be able to go?
So isn't it just because like lactate dehydrogenase like at the end of like glycolysis with like driving pyruvate to lactase? Would that be like inhibited? Or no,
So
it wouldn't be inhibited. That makes
so
sense.
no, you're we're getting closer, Tyler. We're getting closer.
Okay.
Okay. So let's go back to our other example. We're going to walk our way through all of this. Okay. Because I think it's a good example of how everything is in some ways sort of interconnected. I don't have oxygen. I can't run the electron transport chain. Okay. If I can't run the electron transport chain, then what's the pathway or the piece of the pathway that's immediately upstream of the electron transport chain?
The Krebs cycle.
Very good, Jackie. The Krebs cycle. Okay. Remember, we went over in class today that essentially the Krebs cycle is faster than the electron transport chain. And so it's going to be limited in many ways by what's happening in the electron transport chain. So if I can't run the electron transport chain, I can't free up NAD and FAD to go back and allow the Krebs cycle to continue. So if I can't run the Krebs cycle, then go back one step further. From a carbohydrate standpoint, what are we putting into the Krebs cycle from the end of glycolysis?
Pyruvate.
Pyruvate. Okay. So if I can't put the pyruvate into the Krebs cycle, the pyruvate's going to build up, and it's going to build up inside the cytoplasm in our cells. And if it builds up in the cytoplasm in our cells, Tyler, what enzyme might it come into contact with? Tyler didn't hear me. It was the thing that she just mentioned a second ago.
No, I literally did. My brain isn't working right now.
That's okay.
So you
What
said what?
enzyme? If
It's lactate
pyruvate,
dehydrogenase.
very good.
Okay.
Okay. So I come all the way down the Krebs cycle and I'm going to make pyruvate.
Mama.
And then that pyruvate is either going to go into Krebs or it's going to go into lactic acid.
Mm-hmm.
And if it can't go into Krebs because the Krebs isn't running because we don't have any oxygen, so the electron transport chain isn't running, then the only way we're going to be able to make energy then is for it to go into lactic acid. Okay. So it's sort of the same idea behind things. Okay. So the idea is if I can't recycle NAD into NADH, then the same thing, or NADH into NAD, then the same thing happens. It's the same thing happens if you don't have oxygen. That if I can't clear all of those things through the electron transport chain, if I can't donate my electrons at the electron transport chain, then the free carriers can't go back into the Krebs cycle. And if they can't go back into the Krebs cycle won't run. And so therefore, there's no ability to put pyruvate into the Krebs cycle or fat into the Krebs cycle. So what I'm left with then is the only thing that can happen is that pyruvate can go to lactic acid. Okay? Does that make sense, guys?
Yeah, that helps a lot. Thank you.
Okay. But that's the kind of thing where you got to think about like, okay, well, what regulates what and what's going to go where and, all of those things. Okay? Okay. Are there has to be other questions.
There were two questions on the short answers that like were mentioning signals or like signaling molecules, processes, and then the next one after it was what signals. And I just didn't
Sure.
know if you like were you meaning more the like hormones or the like mTOR, all of that stuff. I was just didn't know which parts you wanted.
Yeah, for sure, Sarah. Can you read me the question? I don't have it just
Yeah,
right off the top of my head.
yeah, so one of them is like a three-part, but the last part says you're like explaining to someone that wants to work out.
Yeah.
Explain to them the stimulus or acute physiological change that occurs during and in the hours following each training session that help drive the chronic adaptations. Be specific about initial signals, signaling molecules, and processes.
Okay. So in that context, right, the process is protein synthesis. If I want bigger muscles, I have to run protein synthesis. I have to make more actin and myosin to make my muscles get bigger. Okay, so that's the process in the back end. Okay. Then what is the primary thing? What is the initial? Let's go the other direction then. What's the thing when you go work out today? What are we trying to accomplish? What's the thing that we need to get that's likely going to signal the increase in hormones that's going to help us also signal to activate mTOR, which is kind of the big thing. We want to activate mTOR because the mTOR drives the protein synthesis. So what are you trying to get? Because this should inform what you go to work out with today. Anybody remember?
Is it just a positive protein or
So upstream of the positive protein balance, I guess, Sarah.
okay.
So like if I start doing squats right here in my living room, what is happening in my muscles that will help to increase the release of anabolic hormones and then also by itself begin to stimulate the mTOR in those muscles?
Is it because of like muscle loading and stretch?
That's right, Tyler. It's load and stretch. So the signal, right, is load and stretch. And then the signal leads to some hormones. And then that is then going to lead over to, that's going to then lead to the activation of mTOR. So let me try something. No, I don't want that. I don't want a template. I just want the normal thing. All right, guys, I'm going to try. Maybe this will work. I'm going to share my screen. And then I've got a whiteboard. I'm going to pull this thing off. My pen doesn't have that. Let me see if I can do this. Okay. So if we get load and stretch. Okay. And the load and stretch are going to signal to sort of all by themselves activate mTOR. They're also going to stimulate hormones and they're going to increase. Let's see if I can shrink this. No. That's the other deal. There we go. And then they're also going to increase hormone receptors. I'm sorry, that's terrible. But you work out, you get load and stretch. Okay. Loading and stretching causes the release of anabolic hormones. Load and the stretch also all by itself stimulates mTOR, but then the hormones, it also stimulates increase in hormone receptors, which brings more hormones in, and then the hormones also activate mTOR. And then downstream of mTOR, this is where we get protein synthesis. Okay. That's how that whole process is going to work. So when you go work out today, Sarah, what do you need to make sure that you do to your muscles today if you want to try to drive protein synthesis?
Lift heavy enough that there's like a good amount of load and then do like dynamic stuff so then they're stretched.
Right, and the stretch is going to come through what kind of contraction?
Eccentric.
Perfect. There we go. So
That makes
that's
sense.
why That's the link in those particular things. You have the initial event during a bout of exercise. We want to load them, we want to stretch them. That is going to then, signal the other things downstream that will activate the process that's going to be protein synthesis. And then if you activate it today and it stays elevated for a couple of days, and then you work out again on Wednesday and it stays elevated for a couple of days, and then you work out again on Friday, right? And you do this over a long, long period of time, you end up in a place where chronically you're sort of spending more time in this state of increased protein synthesis. And hopefully that then will allow the muscles that you've been working out that are experiencing elevated protein synthesis to get bigger eventually. In about how long? How long does it take for our muscles to get bigger?
6 to 8 weeks.
6 to 8 weeks. Very good, Karen. Perfect. I'll write for y'all in Ellis's favorite pen color. Okay. But let's remember this. We'll go back to our last question. She loves this rainbow one. We've got glucose, and the glucose comes down. Right. And it makes pyruvate. And then that pyruvate can then become lactic acid, or it can go this way and it can go into Krebs. Okay. And so to me, the easiest way to try to remember and understand carbohydrate metabolism is that while there's always going to be some level of pyruvate going to lactic acid, right? And that's driven by LDH, and this side is driven by PDH. That as long as the pyruvate that we make, as long as it can go here, okay, that gets us more energy, and we don't make anything that's going to inhibit sort of metabolism and further contractions. But the problem is that remember that in order for it to go in the Krebs cycle, Krebs has to be running. And Krebs is controlled by, for the most part, by what happens at the electron transport chain. So sort of the downstream sides of those things then actually control in some ways the fate of pyruvate. And so as pyruvate gets more and more and more, as we make more pyruvate, okay, then there's a greater chance then that pyruvate, if it's not going to go into Krebs, is going to go and become lactic acid. And then once that happens, pH falls, we become acidic, and it inhibits sort of the whole shooting works. Okay? Cool.
Is it okay? So that last question that we're talking about with acute physiological change during exercise, just to make make it clear, because I know you like to have us be specific and the short answers.
I do indeed.
So the initial signals, that would be the muscle loading and the stretch signals, and the
Yes.
the signaling molecules would be the hormone responses and activating mTOR, right? And then the processes would be protein synthesis.
You're 100% right, Tyler.
Okay.
So coming off of that question that you just threw out with the glycolysis, going through the pyruvate and then either going to lactic acid and the Krebs cycle, would that basically explain the two possible fates of pyruvate, right? Which it would either be lactic acid or the Krebs cycle?
Yes, 100%.
Okay, great.
I'm looking. Sorry, guys. I'm looking for my stylus. It may not have any charge, but now it looks like Ellis has taken my stylus and done something funky with it. She loves to draw on my computer. So, okay, y'all may be stuck with me trying to draw with my finger. All right, what else we got, guys?
One of the questions talks about substrate use, like carbs versus fats. So I understand
Yes.
the first part on like fats used, during rest and low intensity, and then you kind of switch to carbs. But then it asks if we could briefly outline the mechanisms underlying the changes. And I don't know like what exactly you mean by that.
So it means why, Victoria, in so many ways. Why are we going to switch? What are the signals that are going to drive the switch from fat to carbs? So why, when exercise intensity goes up, do we stop using carbs or stop using fat and start using more and more carbs? Okay.
Isn't it just because it's like broken down slowly and we need
Mhm.
more energy?
Yeah, that's mostly it. Okay. At least initially, it's just because fat breakdown is slow. Okay. Fat breakdown is going to be slow. And then the other piece of that is, let's imagine, okay, I have Krebs here. I have electron transport chain here. And then I've got, let's just say, pyruvate. So I've got a bunch of carbs over there. And I've got some fat coming from there, right? And so there's this competition, or synergy, or however you want to describe it, where both the fat and the pyruvate are going to get broken down in aerobic metabolism in the same way. Okay. But there's more steps to get the fat in and get it into Krebs than there is to get the pyruvate there. And eventually, when intensity is really, really high, then the amount of pyruvate goes way, way up. So it's not just that it looks like this. The pyruvate looks, basically, I'll try to just draw it and make it even bigger. There's this huge amount of pyruvate. And so what happens then is, because there's so much more pyruvate, that's the thing that is going to encounter and get turned into acetyl-CoA, because there's just so much more of it that is around. Because as intensity goes up, it drives the breakdown of glycogen and it drives the movement of glucose into our cells. And so the pyruvate levels get really, really big compared to how much fat is coming in. And so we just use more of the pyruvate. We need a huge amount of ATP, and we're just going to get it from whatever is kind of the most readily available thing. And that's partially why that happens. Now, eventually, the thing that happens as well is that as we get over lactate threshold, we have more and more pyruvate. We're also going to start making more and more lactic acid. And so remember that those hydrogen ions are going to begin to inhibit fat metabolism, and they're going to begin to sort of inhibit sort of the function of the Krebs and the electron transport chain. And so then once that happens, all that we're left with then at very, very high exercise intensities, we can't break down any fat. All we can really do is break down carbohydrate and make it into lactic acid. And so that's also one of those reasons that during very, very high intensity exercise, we use almost exclusively carbs, is because you become acidic and we can't break down fat. Victoria, does that make sense?
Yes, it does. Thank you.
Okay, perfect.
I have another question. The one that asks about genetic limits. I know one of them is like muscle fiber amounts, because if you have more, then they can get bigger.
Mhm.
But I can't really think of like two more.
Okay. Do you remember the really jacked up cow that I showed you guys a picture
Yes.
of? What was going on with the cow that made the cow so ripped?
He didn't have any myostatin.
Very good, Dylan. The cow didn't have any myostatin, right? Do all of us have myostatin?
Yes.
Okay. Do some of us have differing amounts of myostatin?
Yes.
Yes.
Okay. So that's potentially one, right? If you have more myostatin than me, then you're going to have a harder time making your muscles bigger. Okay. Anything else that we're genetically different on in our muscles?
Fiber types? Oh, not in or
No, that's right, Jackie.
yeah,
Fiber
fiber types.
types. We can argue in some ways, right, that maybe the type 2 fibers don't actually get sort of proportionally larger gains than type 1s, but it's really hard for us to know. But there's a chance that we might, right? Because most people use their type 1s all the time, and so they're already in some ways hypertrophied. So that's a possibility. So it could be fiber type, could be myostatin, could be the number of muscle fibers. Okay. Um, what else could you guys think that might be genetically different between people?
Is having a bigger cross-sectional area the same thing as fiber type?
No, it's not actually, Karen. So bigger cross-sectional area basically means I got a bigger muscle to start with, right? And so that could be an issue. Fiber type is going to be right controlled by the sort of enzymes that are in the individual fibers. So the myosin ATPase, especially, maybe the calcium ATPase. And so there are some distinct things that are going to be there. They might be the same size, but they're going to function a little bit differently enzymatically.
Okay, that makes sense. Thank you.
Would like connective tissue and like tendon strength play a part in that?
Sure, that could certainly be, those can certainly be genetically determined, Tyler, and can very much be different. I don't know to what extent they're gonna facilitate whether or not somebody's muscles get bigger than somebody else. It could certainly play a role in like force production and injury risk and some of those things, but I'm not sure that that's necessarily going to be a thing on that other end.
Would it be having fewer myonuclei?
Sure, yeah. Having fewer myonuclei could definitely be one. Right? We have to at some point add myonuclei. And if we can't add any more, then that's going to put a limit on how much bigger our cells could get. Is there something else that kind of looks like a myonuclei or something that might have played some role in this?
Could it be the amount of satellite cells that you have?
Yes, Sydney, very good. Satellite cells. So I don't know to what extent, if you have more satellite cells, that it makes you get bigger, but we do know that if your satellite cells don't work properly, like if we kill off satellite cells, we can certainly blunt or almost completely block how much bigger your muscles are going to get. And so, is there likely to be some critical amount that you have to have, or you're not going to get any bigger muscles? Probably. Okay. But that's a very plausible thing that we could say. So I think we're well over 3 now. So we're in pretty good shape there. What else we got, guys? Anything? Is that it? Things that aren't necessarily like specifically in a short answer question?
I'm guessing you want us to know like just like the definitions of like biosynthesis and that kind of stuff that's like in the beginning of the slide, right? Okay.
Okay, I have a question.
Yeah, Montana.
This is kind of like confusing to ask, so let me know if it doesn't make sense. But in your question of explain the two possible fates of pyruvate once it's been formed via glycolysis,
Uh-huh.
and you also said what are the advantages and disadvantages of each fate. So I found advantages and disadvantages of whenever oxygen is absent, because obviously lactic acid. But I couldn't really think of any disadvantages for being converted to acetyl CoA and being used in the Krebs cycle. I could only think of advantages.
Okay. Anybody else come up with what you think might be a reasonable disadvantage?
Is it because it's dependent on oxygen?
Okay, I mean, sort of, but not exactly. No, I'm being a little coy.
I just said because it was slow.
Perfect, Sarah. It's slow. That's the real disadvantage is it's slow. Right? There is some limited capacity of it and it's slow.
Could you explain how the creatine phosphate resynthesis has to do with aerobic metabolism? It's not a question, but I was just looking.
So, real talk for a moment, guys. Who's lost? Who have I confused the shit out of with metabolism? I feel like that's where we are.
I'll admit I'm partially lost, but I think if I like go through, because I haven't had a chance to go through that bit with my review, I think I can figure out, and if I can't, I'll email you and try and get some more answers.
Right?
I think for me, just nothing's flowing. It all just seems like random bits of information, and I don't know how to make it like flow together, if that makes sense.
Okay. Sure. You understand the individual pathways, Haley?
Sorry, say it again.
You understand what's going on with each pathway. You just don't know how they fit together, or what are we
Yeah.
lost on?
Like, I think I'm understanding the pathways. It's just how they all like go together and like I don't know if it's like the big picture or
Okay. Okay. Are any of the rest of you guys even here? You just got your computers on, listening. Should just shout nonsense words. What, Tyler?
I think this review helped a lot.
Okay.
There's like the tiny little details where I'm like, is this influenced by this? But it ain't through that whole.
You know, I think on some levels y'all will be, I would say, underwhelmed, but probably relieved at the somewhat more straightforwardness of the questions. I think y'all are making this, you're kind of psyching yourselves out a little bit and making this more difficult than it probably has to be. Right? You know, realistically, if you sort of know what goes in and you know what comes out and you know roughly how much energy you get and you know roughly which ones are going to predominate under which circumstances, then, you know, I'm really not going to ask a lot of questions about how are they all working together necessarily. I just want you guys to know that they are working together.
This definitely helped me a lot with connection, but I did also have Dr. Bin last semester, and
Okay.
he started off with metabolism. So this helped me understand it a lot better than he did because it was basically just memorization.
Okay.
Your office hours are from 10 to 11 a.m. tomorrow, right?
Yes, they are. They are almost already all full with folks that are coming, but please feel free to run by. My guess is if there are multiple people that need to come and have questions, that most of y'all won't care that much to listen and hear the answers from other people. And what I say to one may be in some ways helpful for another one. So, but yeah.
Perfect. Thank you.
Mhm.
And this doesn't really matter, but I'm just curious, how like split are the questions between like resistance and metabolism? Is it pretty like 50-50?
You again presume, Tyler, that I've made the test out yet.
Never mind.
So it'll be, I hope it'll be pretty close to 50-50, kind of one to the other. I don't know. Part of this, I have some things that I know that I want to ask. I just need to pull them together. But then, the other part sometimes with this is I kind of like to get a sense of when people were doing this and when people are coming in to office hours and stuff to try to figure out like what are we getting, what are we not getting? Should I be asking more things over here, more things over there? Like what am I, is there I don't it's not my intent to basically ask you guys a whole bunch of things that you don't know. And if there's some stuff that it seems very clear people aren't quite capturing, then we'll see. So it's going to be pretty close to 50-50. Cowboys just kicked a field goal if anybody cares.
I had a question.
Of course, Haley.
So, like I like to watch YouTube videos, but I feel like if I look up some of these pathways, it's going to be way too in depth than
Mhm.
what you're asking us. So, would that just like not be a good thing to do?
I don't know. I don't have a good answer for that. I think that's a thing you kind of have to, and I'm not trying to be like dismissive of it, but I don't, I don't know. I think it would probably depend upon what the YouTube video was and what you were trying to, like, what are you trying to understand from the video, right? That, is going to lead you to a video where there's too much stuff or there's too much detail or something like all of that. I think that's probably the part that's that it would be helpful for me to know so I could try to give you some directions or that I could just try to actually go back through and explain things to you yourself.
Okay.
So I know that's a terrible non-answer, but that's
No, you're good.
okay. Anybody else? This is your chance.
I see that potential energy says it's like an energy associated with substance, structure, or position.
Yeah.
So kinetic energy, I'm guessing is like, I guess if you're moving, that's just energy then. But is potential energy, so if you're like standing still, is it like the energy? Is that what it means? Or
So
that's wrong?
no, that's a good question, Skylar. So in the context of the stuff that we've done, realistically, potential energy is like this energy stored in carbohydrates and the energy stored in fat and those things, like energy that is in the substrate that we could break it down and realize, but that we haven't yet.
Okay, that makes sense.
Anybody else? Anybody else just have a dog or a cat you want to show us? Or a bird or a fish or I don't know, something?
This isn't really a question over the test, but uh one of the people who weren't here, she told me to like let her know of like I told her that this was going to be recorded because she wasn't going to be able to watch it, but I told her I'd let her know if anything like urgent, like if she something that was going to come up before the test needed to be addressed, and like is there anything like that that we should know about?
I I I'm not 100% sure what you're asking, Jasmine. Like just like I'm gonna post the recording of this. It'll be on the YouTube channel tomorrow morning. Just like today's lecture is already up. I wanted to make sure that you guys had all of that. So, but I mean, I don't know about anything anything urgent. Um, I'm not like if that person has a particular question, then they can certainly ask, or if they can tell you, then we can try to answer it. But I'm not sure. You know, there's no new material or anything, so I'm sorry, I'm not 100% sure how to answer that question.
Okay, I was mostly a yes or no. If there's like anything that we need to know, before the test came up, or like, they didn't give me any specific questions, so I thought I'd ask.
No, I'm you're a good friend for doing that. I mean, there are lots of things that they need to know, but I don't know that there's anything, sort of brand new that we're just revealing in the midst of this in comparison to anything else.
In the energy stores, like the pathways,
Mhm.
so like the immediate ones, I guess they're used first. So like ATP, creatine
Yeah.
phosphate, and the myokine, those are used first. I'm guessing what's used second is the non-oxidative ones, and then
Mhm.
oxidative third.
I shudder when we try to say it like that, Skylar, but because we're still getting some from all of them all the time, but that's a generally reasonable way to try to say like which ones are we getting most of our energy from, right? If you're doing something kind of at the very start of exercise, you're going to use more so you're going to use the immediate ones, and then you're going to use glycolysis, and then you're going to hope, right, that as those begin to run out or as you begin to make a little bit of lactate, that you're going to get your aerobic metabolism kind of warmed up and ready to go to kind of take over for those two so that you don't continue to make lactic acid and you have enough ATP to be able to continue to exercise.
Okay.
For the protein balance, on the short answer, I'm a little
Mhm.
confused how like exercise would push muscle fibers to like a positive protein balance state.
So, Victoria, that goes back to one of the first questions that we answered. And so, right, you gotta have to sort of work backwards from all of that. So, what is it that we need to drive our positive protein balance? We need to activate mTOR in the muscle. We need to activate the mTOR pathway, and we need to make sure that you've eaten some protein, some amino acids that are going to be available. And so you got to work backwards from there to figure out what is it specifically about exercise that leads to the activation of the mTOR pathway. And that's where load and stretch and some release of anabolic hormones and those things in response to that bout of exercise are going to come into play. So that's what you're trying to achieve from a bout of exercise is that. And then you got to make sure that you're eating enough. And so that when that pathway gets activated, there are amino acids available to help make those new proteins, which are going to be acted in myosin to make your muscles bigger.
Okay, thank you.
Anybody else? Going once, going twice.
Whenever pyruvate after glycolysis is whenever it goes to lactate, is that so that's anaerobic, but is it used faster than it is if whenever you go to the aerobic side and go to the Krebs cycle?
So getting from glucose to pyruvate, that takes just as long, right? No matter what happens, Skylar, because it's the same steps, you end up in the same place. But what happens is when you go to lactic acid, the big thing that it lets you do is it allows you to take the NADH that you formed right as it split from glucose into whatever it is, the first of the 3 carbon molecules, which is whose name I have forgotten, and it lets you recycle that. And so it allows you to keep glycolysis going, basically, by doing that. And so in that way, it keeps the pathway going, and it's faster because you still have to take the pyruvate, go all the way through Krebs, all the way through the electron transport chain, which is going to take some amount of time before you can realize all of that extra energy. So, even in the grand scheme, if all of that extra energy takes an extra second or something to make, if you're doing very, very high intensity exercise, that 1 second matters. If you don't have enough energy to continue at that, then that can become a problem.
So whenever it goes to the lactate and then it just goes back through glycolysis, you said?
Mm-hmm.
Okay. And then aerobic, it goes through the Krebs cycle and it's used like long term.
So, you may use it right there, but it's just going to like right in that moment, but it's just going to take longer for it to kind of realize that.
Oh, okay.
Realize that the ability to get that extra energy, if that makes sense.
Yeah, that does.
Anybody else? Okay. Well, I should be around most of the day. Tomorrow, if you need something, drop by, stick your head in, say hello, say I'm confused, Dr. Black, whatever it needs to be. I got to step out for a little while. I have a meeting with the ROTC commander, I think at 1, so I'm gone from like 1 until 2:30, and I've got quite a few of you guys that I know have already kind of scheduled some meetings to come through. But if you need something, reach out, let me know. Otherwise, I will see you bright and early Wednesday morning.
Thank you.
See you Wednesday. Thank you.